Hello and welcome to this Nature Research custom media webcast titled Network-Anchored Functional Genomics Identifies Intratumoral Axes of Vulnerability in Pancreatic Cancer. My name is Sarah Hiddleston and I will be your moderator. Today's webcast is sponsored by Selecta. We'll begin the webcast with a joint presentation from two scientists in the Drietta Lab for Functional Genomics Research at MD Anderson Cancer Center in Houston, Texas. They are Johnathon Rose, Graduate Research Assistant, and Sanjana Srinivasan, Graduate Research Assistant. This will be followed by a short overview of Selecta from Dr. Paul Deal, Chief Operating Officer at Selecta. We'll then move on to a question and answer session with all the speakers. You can ask a question at any point you wish throughout the webcast. To do so, please type your question in where it says type your questions here, and then press submit, and we will answer them today. And now, over to our speakers, Johnathon and Sanjana.
So, hello, my name is Johnathon Rose.
And my name is Sanjana Srinivasan.
And today we're going to talk about our project where we have integrated transcriptional diversity and functional genomics to inform on intra and inter-tumor heterogeneity within PDAC. So pancreatic ductal adenocarcinoma is a highly lethal disease. It's the most common form of pancreatic cancer with a very poor survival rate. And it's en route to become the second highest cause of cancer-related death by 2030. So the current standard of care for this disease provides only a marginal benefit to survivability, and there's really a limited opportunity in terms of therapeutic intervention for this disease. So with this in mind, one of the foundations of what we're doing is called PILOT. So this is patient-based in vivo lethality to optimize treatment. Originally developed by Ali Carugo, this allows us to perform custom RNAi and CRISPR screens in vivo setting using pre-prioritized libraries. And with the idea of first establishing in vivo engraftment using empty barcode libraries, applying our custom libraries in green, which are annotated based on multiple genomic and genetic features. And then once we identify targets of interest, we moved it into the PDAC for preclinical annotation and pharmacological inhibition. So one of the early manuscripts published in 2016 by Ali Carugo highlights utilizing this approach to identify and inform on WDR5 and its association with MYC as a critical determinant in pancreatic cancer. And this is an original screen performed using an epigenome library of 230 genes, whereby we're able to identify WDR5 and other targets as an oncogenic dependency, and subsequently reinforce this with the clinical relevance, establish its role in tumor maintenance, rescue the phenotype, and of course, compare survivability. So along with this, we also expanded using a custom surfaceome library, which we developed in-house to help identify Synakin-1 as a critical mediator of necropinocytosis in pancreatic cancer. And this is something that I'll be talking about a little later and how we've used this in the human system. But for this specific manuscript, we focus on mouse cell lines derived from the KRAS mouse model. And overall, this approach helps us to identify essentially genetic targets, but doesn't necessarily help us identify the true context of these targets every time. So in order to address establishing context dependency, we have to understand heterogeneity in pancreatic cancer. And heterogeneity in pancreatic cancer, and specifically oncogenic signaling diversity in pancreatic cancer, is driven by multiple facets. And even though pancreatic cancer has a relatively flat mutational landscape, primarily driven by the big four mutations, KRAS, P53, and so on, there are less frequent mutations that occur in the tumor, as well as ongoing epithelial mesenchymal transition that can essentially dramatically affect the dependency spectrum of these models and of these tumors. And it becomes very challenging that when we identify a target of interest using a custom library approach, to take that and actually apply it to the clinic and apply it to our preclinical models as well. So with this in mind, Sanjana and I established a system in place. And there's some information in the point, and this is actually an important method, because even with recent advancements showing how germline BRCA mutations can be used to stratify patients for maintenance therapy with PARP inhibition, that alone has shown some significant success. So if we're able to take that and expand on it, then we have an opportunity to really try and make a clinical impact with these targets that we identify using our functional genomics approaches. So with this in mind, Sanjana and myself applied a two-prong approach to help quantify PDAC diversity, both in the larger transcriptomic context and also in the functional context. The transcriptomic context being on the left arm and the functional being on the right, and ultimately integrate these two to help basically inform on context for these individual targets. And this is essentially a data-driven platform to identify, signatures that contribute to PDAC diversity and anchor gene targets within these signatures. So with that, I'll pass it on to Sanjana.
Okay, thank you, John. So the first thing that we did was utilize a large cohort of patient-derived xenografts, or PDX models that we have. So PDAC in general is broken down into two major subtypes, so classical and basal-like, with the basal-like tumors showing overall worse prognosis and poorer response to treatment. So we have about 50 PDXs that were developed with IAX and traction within MD Anderson that we utilized for this study, and they range from classical and basal-like, and all of these on all of these tumors, we collected mutation profile and RNA expression. So first, rather than just looking at differential expression that's driving these different tumors, what we did is we wanted to capture the overall diversity of transcriptomic signaling that we see across these tumors. So to this end, we collected pairwise correlations of all dynamically expressed genes within these tumors, and using a log likelihood refinement method, refined this to gene pairs that were highly likely to be biologically relevant, and then developed a network out of this and used a random walk algorithm to break this down into various clusters. So what we get out of that is this PDAC co-expression network, or PSEN, which is comprised of about 7,000 genes and 31 clusters, some of which, as you can see, have some Go annotations for biological relevance, and some of which are still pending annotation. But overall, what this is this is a network, this is a platform for us to be able to quantify signatures that are contributing to disease diversity within PDAC. So what's of note here is that this is a force-directed layout, meaning that each dot here, which represents a gene, the closer they are localized together, the more correlated they are not just to each other, but to the neighborhood in general. And conversely, the farther apart that they are visualized, the more anti-correlated they are to not just each other, but that whole neighborhood in general. So what we were first interested in was looking into what these strong anti-correlating signatures are. So we identified two separate sets of signatures. The first one being between cluster 1 and cluster 23, where cluster 1 was enriched for processes involved in lipid metabolism, and cluster 23 was enriched for cell development. And conversely, cluster 2 and cluster 13, where cluster 2 was enriched for Golgi vesicle transport, and cluster 13 was enriched for nuclear transcribed mRNA catabolism. So when we looked into these clusters specifically, we were interested in looking specifically into the cluster 1 and 23 anti-correlation. And the reason for that being all of the known classical genes that were previously described were unbiasedly recapitulated within cluster 1, which is the lipid metabolism cluster, and a large proportion of the basal-like genes were unbiasedly clustered within cluster 23, which is the cell development cluster. So we developed a module centroid method where we're basically getting a centroid expression of all the genes corresponding to each cluster. And when we look at our models within the PDXs, we see that while the classical models are not necessarily overexpressed in cluster one, there is a large gradient of depletion that we see within cluster 1 within our basal-like models, suggesting that this isn't necessarily a binary classification of classical and basal-like, but rather that it seems to be more of a continuous transition. So to look into the signature a little bit closer, I'll draw your attention to the heat map in the center here. What we did was look at the centroid score for cluster 1 and 23 across all 50 of the models that we used in the study. And what we see is a near perfect anti-correlation where if cluster 1 is enriched, cluster 23 is depleted, and conversely, if cluster 23 is enriched, cluster 1 is depleted. So this bar graph on the left here, denoted by Moffitt, shows the Moffitt signature subtype of classical versus basal-like, where we see that most of the classical models are enriched for cluster 1 and the basal ones are enriched for 23. However, there's this group in the middle that seems to be partially expressing both cluster 1 and cluster 23. So when we apply k-means clustering to this, we're able to separate out this middle group, which we're calling the quasi-basal group, that seems to be expressing both classical and basal-like phenotype at the same time. And when we look into clinical histology and recurrence, we're also able to find that while the quasi-basal models have slightly more tumors that have poor differentiation and more distant metastases, when you look at the PSEN-derived basal models, you're able to capture a set of tumors that are showing completely poor differentiation and distant metastases. And on the far left, when we look at the expression profile of all of the genes within the PSEN, we're seeing that PATX90, which is our most classical model, is showing a general enrichment for the entire cluster 1 axis and depletion in the cluster 23 axis, and conversely PATX148, which is our most basal model, is showing almost complete depletion in the cluster 1 axis and enrichment in cluster 23. So this is just showing that it's not one or two genes that is driving the signature, but this entire cluster and this entire axis that seems to be changing in their expression signatures as the tumor goes from more classical to more basal. And on the far right here, when you look at principal component analysis, it seems that the subtypes are the most salient feature driving the PCA. However, when you include the quasi-basal group on the right, you can see that it's perfectly nestled between the classical and the basal-like tumors. When we take the PSEN-derived classical tumors and the PSEN-derived basal tumors and we do differential expression, what we're seeing is the most salient signature we see coming out of this is EMT, suggesting that epithelial to mesenchymal transition seems to be what's driving a tumor from more classical state to a more basal state, which we can all appreciate is not a binary transition, but rather a continuum, which we're able to capture with the PSEN method. We're also able to recapitulate this with patient tumors. So this is looking at the TCGA data set of high purity PDAC models. So even here, we're able to see that you're recapitulating a near perfect anti-correlation between cluster 1 and 23 enrichment. And again, we're showing this large group of tumors that are expressing both cluster 1 and cluster 23 at a partial level. And when we look at differentiation status, when we pair this with Moffitt's two subtype system, you are seeing an enrichment for more poor prognosis, poor differentiation tumors within the basal. But when you use the PSEN method, you're again able to pick out tumors that are 100% showing poor differentiation status. So this really begs the question for us of whether the quasi-basal group, when you're looking at it at the bulk scale, if this is a result of competing signatures that you're seeing from single cells that are both classical and basal, or if on a single cell level as well, you're able to capture cells that are transitioning from classical to basal and are in the quasi-basal state. So to address this, we performed single cell sequencing on 3 cell lines that we derived from our PDXs. So PAT269, which is our most classical cell line, PAT124, which was quasi-basal on the bulk state, and PAT53, which was basal in the bulk state. And first off, what's of note is, as predicted, PAT269, which is most classical, has a large proportion of single cells that are classical, and PAT253, which is most basal, has a large proportion of single cells that are basal. However, when you look at PAT124, you can see that it's not a result of competing subclones that are both classical and basal, but even on a single cell level, you're seeing cells that are transitioning from classical to basal. So a bulk signature of quasi-basal denotes that even on a single cell level, the single cells are also quasi-basal. And another thing to note here is that regardless of the bulk subtype that we're seeing, the largest proportion of single cells that we're seeing seem to be falling within this quasi-basal subtype. So even on a single cell level, even within classical and basal tumors, you're seeing a large proportion of cells that seem to be transitioning from one state to the other. So having characterized the transcriptional diversity that we see across PDAC, we then wanted to look at we wanted to understand the functional diversity that we see within these tumors. And to address that, I will give it back to John.
So along with helping understand the transcriptomic signatures that underlie PDAC diversity, another component that we wanted to explore is essentially heterogeneity and oncogenic signaling in pancreatic cancer. In order to do this, we applied a top-down in vivo functional genomics platform to start to quantify oncogenic signaling diversity starting from the cell surface. And for this, in a similar vein to the Silicon-1 manuscript where we applied mouse lines, in this context we applied early passage PDX lines, the same PDX lines that we ran single cell sequencing on, in order to model this human diversity. And we started by selecting a pre-prioritized PDAC surfaceome and generating a custom library for this. And then we expanded on this using CRISPR, a custom CRISPR library that not only annotated the functional surfaceome, but also expanded downstream and along the surface as well based on the string protein-protein interaction network. And in this way, we can start to look at oncogenic targets in the context of other oncogenic targets and not just sort of in isolation. Along with this, we also ran a parallel screening methodology, which allowed us to compare in vitro and in vivo settings. And essentially, what we did is, upon establishing a reference population, following transduction and MOI confirmation of a pooled lentiviral library, we ran parallel time points in vitro and in vivo, and also in vivo was conducted orthotopically for these human PDX lines that we've discussed previously. And ultimately, what this allows us to do at the bottom right is compare against the original reference population and identify barcode depletion and barcode enrichment. And in doing so, you'll see I have a plot here on the x-axis. You identify in vitro, and on the y, you see in vivo. And what this lets us do is identify targets that are in vivo enriched for every PDX model, targets that are in vitro enriched, and targets that score across both. Now, what's interesting here and why I wanted to show this is that the rank order of your hits, and even hits the rank order of your hits changes dramatically based on the context of the tumor modeling. So if you model in vitro, you can have in vitro specific targets. And if you model only in vivo, you can have in vivo specific targets. And this is important because we don't want to just be focusing on things that only score in vitro, for instance. And based on this, we performed in vivo screening across all 4 models and took this information and looked at our models from most classical, which is PATI69, to most basal-like, which is 153, and started to realize that, there's only really a few targets that score across everybody, and these guys are ERBB2, 3, and 4, for instance, along with some more novel targets that we're also exploring. But overall, the realization here is that it's going to become challenging to just take a target and essentially understand the mechanism of that target and apply it to pancreatic cancer as it is, because of how diverse these tumors are. So with this in mind, that was what really drove us to expand on these original findings and apply a CRISPR library, and using a string PPI network to expand on these functional RNAi-defined hits. On one hand, you are able to sort of double down on these original findings that we found in RNAi using CRISPR, and on the other, we're able to expand around these hits as well. And what this lets us do is build sort of a network of hits for each model and merge these networks across each other and see what's going on. And that's what this network on the left here is. Essentially, these are 3 merged dependency networks, where on the top right hand here is the methodology where we pre-prioritize the PDAC surfaceome. We annotate this early on using RNAi. We then expand it at the bottom and build up these networks. And then something else that I should mention is that we had to apply an in-house methodology for in vivo functional genomics for analysis, which we call low-fat bagel. And just for visual here, the more positive targets are more essential, the more negative targets are less essential, and the more positive targets are here are in red, and the more negative are in blue. So you can see PATI69, for example, is a very classical model, and you can see SMAD4 is in blue right here. And as I go from PATI69 to PATI124, you can see SMAD4 drops out. We suddenly have, even though we have similar central targets, we have a complete shift, well, not a complete shift, I should say, but a shift away from 69 targets, more around RTK signaling. And then I go again to 53, the basal-like, and there is a very dramatic shift. SMAD4 is now red. And we can see the importance of understanding these models in the context of our network signature, because suddenly there are vulnerabilities here that we would have never explored, and this is going to help with stratification. So this is essentially the 2 arms of our approach, and ultimately our goal here is not only to sort of define them in isolation, but to integrate these 2 approaches. On one hand, using transcriptomic signatures to define diversity in pancreatic cancer. On the other, starting from the cell surface, expand on heterogeneous signaling downstream, and anchor these targets within these signatures themselves, especially against prominent anti-correlating signatures that we also match against other featured subtyping strategies like Moffitt. And that's what we have here. So on the top, we have our string PPI networks for PATI69, 124, and 53, and below we have our targets anchored within the network that are color coordinated. And what's interesting here is that you can see that there's a clear trend in targets, especially around 53, which I will focus on over here, around that cluster 23, around the cluster 23 and all of the clusters interconnected around cluster 23 as well, which is interesting. But the next thing we needed to do was validate this. And to do that, I'll hand it back over to Sanjana.
Thank you, John. So what we wanted to do is to understand the signatures that we're seeing specifically within cluster 23 and how these relate to dependencies that we're seeing that are anchored within cluster 23 within the basal models. So to address this, what we did was we performed single-cell sequencing using a 10x feature barcoded platform. We knocked out 3 genes, ILK, SMAD4, and ZEP1, that are anchored within the cluster 23 axis and were only hits in PATI53, our basal-like model. We knocked this out in our classical model and our basal model to look at what the impact was on the transcriptional heterogeneity of the tumor that we see. So we, like I said, we performed this with ILK, SMAD4, and ZEP1, which were 3 basal hits, and ABCG8, which is a negative control. And on the left here, you can see the UMAP for PAT69. There's no real difference between any of the knockout conditions and the negative control, suggesting that knocking out these three genes did not have any impact on the transcription of the surviving cells. However, when you look at PAT53 on the right, you can, which is our basal model, you can see that the red dots here, which are the negative control, ABCG8, are all clustered together on the left, compared to ILK, SMAD4, and ZEM1 knockout cells, all clustered together here, suggesting that there's a stark transcriptional difference between the negative control in this condition, in this cell line, and each of the knockout conditions. So when we look at this a little bit closer and overlay it with our cluster 1 and 23 anti-correlating signature, what we see is on the left for PAT69, within the negative control, you're seeing a large peak for a large population of cells having a quasi-basal phenotype, which is something that we showed previously with the unperturbed PAT69 cells as well. However, with PAT69, for each of the three knockout conditions for ILK, SMAD4, and ZEB1, you're seeing a loss in the percentage of cells that are falling within this quasi-basal state, and you're seeing a gain in the number of cells that are falling within the classical state, suggesting that the knockout of these three genes is not just impacting basal population, but it's also causing a reversion of quasi-basal cells to a classical state. Compared on the right to PAT53, we can see that the negative control has a large population of single cells that are basal and also quasi-basal. And in each of the three knockout conditions here, you can see that there's a stark change in all of the basal cells, where there's almost complete death of all single cells that were previously basal. And in all three cases, what you're seeing on there, not much of an impact with the quasi-basal population, but what you're seeing on the right is an emergence of single cells that are actually classical, suggesting that not just suggesting that basal cells are dying as a result of this knockout, but that quasi-basal cells that perhaps harbored partial dependency towards the basal conditions. I'm sorry, let me repeat that. What you're seeing with PAT53 is not just that each of these knockout conditions are killing the basal cells, but they're causing quasi-basal cells to revert back to a classical state, suggesting that these quasi-basal cells that perhaps harbored a partial dependence on these basal genes are reverting back to a classical state in order to survive. So this is the first time that anyone has genetically validated these subtypes and shown that the subtypes are more plastic than previously thought, and we're able to actually, using genetic perturbation, impact the clonal dynamics of single cells within a cellular population. So with that, we will conclude. So just as a summary, the PSEN, the PDAC co-expression network, provides a novel and quantitative approach to capture transcriptomic signatures across various different biological processes to help shed light on intra-tumoral heterogeneity within PDAC. Using the top-down functional genomics approach that we have developed within the lab, we're able to further characterize this heterogeneity using surface protein dependencies and downstream targets to understand context-specific vulnerabilities. And we're also able to show that prominent cluster 1 and cluster 23 anti-correlated signatures are able to more granularly recapitulate known subtypes and shed light on the fact that these are not binary subtypes, but rather a continuum that can be affected. And finally, using the feature barcoded single-cell RNA-seq system that we have, we're able to genetically validate some of these genetically validate the subtypes, and also show that the subtypes are plastic and can respond not just by cell death, but cellular adaptation to different knockouts. And with that, thank you to everyone that has helped us with this project, our PI, Julio Draeta, and specifically Ale Carugo and Tim Heffernan as well. And thank you to all of our funding sources, specifically the CBRIT Graduate Scholar Program that funded me throughout my PhD, and the Paula Altman Goldstein Discovery Foundation that has funded John Rose. And thank you.
Thank you, everyone, for tuning into the webinar today, and thank you, John and Sanjana, for sharing your experience using our CRISPR technology to investigate adaptive drug resistance in pancreatic cancer. Before we take questions, I'd like to give a very brief overview of who Selecta is and what we do. We're a small technology-focused company just south of San Francisco, Mountain View, California. We're not a venture company. Our company started about 12 years ago with seed money from research grants raised by our founder. And we use that money to develop technology for products and services for functional genetic screening to help identify genes that are responsible for disease progression, drug resistance, cell differentiation and development, and other biological processes. And in particular, the goal was to create technology platforms that enable broadly parallel simultaneous analysis of large numbers of genes or genetic elements at once, a genome-wide type of analysis. Our goal is not to develop technology that requires large-scale automation, limb systems, microtiter plates where you're doing, large numbers of parallel assays in single wells. It's really to make this technology accessible to general labs by providing tools that can be used by a wide range of researchers using basic laboratory techniques such as molecular and cell biology. The way we've done this is by leveraging the rapid development of next-generation sequencing technology over the past decade. Next-generation capabilities have increased so that it's able to be used to deconvolute results from complex biological assays and measure simultaneously many thousands of assay elements in a single reaction. So using NGS as a readout makes it possible to pull results out from thousands of individual reactions at once and track the changes in multiple samples, treatments, different time points, etc. We've had some success with this approach. Here's a sample of some of the publications over the years that have cited use of our technology from customers or groups we're collaborating with. And we've had a more complete and updated list of these citations on our website. The link is shown on the slide. I'd encourage you to take a look at that site, and it has a search function. If there's some particular application, cell systems, or procedures you're interested in, you may be able to find something relevant that will help you with your experiment. The first platform I'll talk about today is the driver map expression profiling assay. This technology enables you to get data on which genes are activated or deactivated in different samples from different treatments, from cell culture conditions, biopsies, other patient-derived material like blood. Probably the most common approach to look at changes in gene expression on a global genome-wide scale is RNA sequencing. That's probably what most researchers are familiar with. And while that's a very powerful approach, it's also somewhat technically complex, with some challenges in prepping and running the samples, and also since it requires reverse transcribing the entire transcriptome, data analysis can be intensive. You really need to establish a robust pipeline involving a team of individuals with different specialties to effectively run RNA sequencing on large numbers of samples and actually analyze those. For expression profiling on a small number of genes, however, the gold standard is still really qRT-PCR. It's a very straightforward and quantitative technique, and it gives you robust and accurate measurements of each targeted transcript. The problem is, though, with qRT-PCR, you can really only look at a handful of genes. With the driver map platform, we've combined the simplicity and sensitivity of qRT-PCR, but on a more global genome-wide scale that gives you comprehensive data, similar to what you would get for RNA-seq. We were able to successfully do this by developing a set of 38,000 primers, 19,000 PCR primer sets, that target all 19,000 human protein coding genes in a single multiplex reaction. As a result, you start with total RNA, since it's a targeted reaction, and you can run one single reverse transcriptase and then PCR amplification and get 19,000 amplicons, each unique for a different human gene. And that then can be, those 19,000 amplicons can be quantified by next generation sequencing. The advantage of the approach is, since it's PCR-based, it's very sensitive. It provides robust genome-wide profiles from a very small amount of sample, down to picogram levels of total RNA. And we routinely run this analysis on samples with just a single or a handful of cells. Also, while 19,000 amplicons may seem like a lot to sequence, it's much less complex than the transcriptome that you get in RNA sequencing analysis, the fragmented transcriptome that's produced with that technique. And as a result, we get deeper sequencing and stronger reads of low-expressed genes with the driver map approach, with 5 times fewer sequencing reads than you would see with RNA sequencing. So this is RNA sequencing shown on the right hand of the slide, and then the results from the driver map approach with only 5 million reads shown on the left. It's very easy to run also. It's a single RT-PCR based reaction with targeted primers. So you can start with total RNA. There's no mRNA enrichment and beta globin depletion. Also, the analysis is straightforward. 19,000 amplicons are defined. They're defined amplicons, so we know what sequences are being amplified. And so they can be easily aligned with the expected reference sequences. And the read levels for each one of those amplicons directly correlates to the expression level of a gene. As a result, you can do the analysis really on a spreadsheet. Depending on what you're interested in understanding from a system, then the driver map can provide a much more straightforward and effective approach to profile all protein coding genes in your samples in a much easier reaction than qRT-PCR. Than RNA sequencing. While driver map and other expression profiling techniques provide an understanding of which genes are up and down regulated, these approaches do not tell you really which genes are important for regulating or driving the changes that you're looking at if you're looking at particular phenotypes. Which activated or deactivated genes actually cause the biological changes of interest? To address that question, you need to disrupt or perturb each of the target genes and really look at the effect it has on what phenotype you're looking at. RNAi and CRISPR are really the only 2 technologies that enable this sort of gene disruption to be done on a whole genome level. With both of these approaches, you can design a small RNA molecule, shRNA or short hairpin RNA for RNAi, or sgRNA for CRISPR, that can be used to target genes across the genome, and you can then introduce a library of these into a population of cells and disrupt or otherwise perturb different individual targets in different cells across the population and look at the results. The CRISPR pooled screening approach then is a very effective approach to identify genes responsible for driving biological changes by looking at them, the effects of disruption of genes across the population of cells and seeing how that affects the growth of the cells depending on which genes are up or down, which genes are disrupted. With any CRISPR screen, the approach is basically the same. First, after making the library, it's packaged as lentiviral particles that are then transduced, in other words, infected, into target cells, and then done at such a level that you get each of the guides in a large number of cells, and several hundred cells in the population. However, you do it at a low enough MOI so that most of the cells in the population only pick up one of the sgRNA. So only one of the genes are disrupted in each one of the cells. If you're looking for an essential gene, then the RNA that are targeted, by the gene will be toxic to the cells. What you're looking for is genes that, when they're disrupted, are toxic. And these cause the cells to die over a period of time. And so after a period of time, the sgRNA targeting the essential genes will be underrepresented in the population. This is a reverse or negative selection, and this is probably the most popular type of screening approach. The depletion of the sgRNA in a population after a period of time is detected by NGS after isolating the genomic DNA from the cells. Presumably, then, each one of these is targeting genes that are essential under whatever conditions you've grown the cells in. For example, if you added a compound, they might tell you which genes are responsible for resistance to the compound of the cells, or other vulnerabilities depending on what you're looking at. Other screening approaches, however, are also possible. You can also treat cells with a compound or factor that will kill most of the cells or is toxic to most of the cells, and identify genes that disrupt that effect of that compound or factor. So you may be able to find genes that are important for the mechanism of action of a drug, or that are important for, say, apoptosis or some sort of effect of cell or arrest of cell growth. This can also be done with a FACS type of screening too, where you're sorting populations of cells and looking for ones that are overrepresented by expression of a particular reporter or activation or deactivation of a particular pathway. The technology that we use to make the sgRNA and shRNA libraries is not limited, of course, to those type of molecules. Really, we can make large libraries of various DNA sequencing using the same cloning tricks. So with a defined pool of oligonucleotides, we can construct lentiviral libraries that have millions of defined sequences or barcodes. And then these can be used to be transduced into cells and uniquely label hundreds of thousands of cells in a population. Since the barcodes are on lentiviral vectors, they're integrated into the genome and they're heritable. So you can create a founder population of cells that, as they propagate, all the progeny from that founder population then also have the barcodes too. This type of labeling can be useful in a number of applications. One of them, as presented in our last webinar by colleagues of Sanjana and John at MD Anderson, used this approach to look at cell heterogeneity and subpools in the population. For example, to look at how the population of cells change over time with differentiation, implantation, etc. On a single cell level, with single cell platforms such as a 10x system, genome barcodes in the cell can also be correlated with the activity of particular genes or changes in specific expression profiles, as was touched on with the presentation earlier in this webinar. It's possible to add sgRNA into the mix and look at how different subpopulations respond or are created when certain genetic changes are purposely introduced. This is related to sort of the perturb-seq or crop-seq approach that many of you may be familiar with. So with this presentation, I just wanted to kind of give a quick overview of the technology that Selecta offers. If you're interested in discussing any of these platforms or approaches further, or get more information about products or services that we may have available, please feel free to contact me. Again, I'd like to thank you for tuning into the webinar, and I hope you found it interesting.
Hello. Thank you very much, Paul, and thanks to Johnathon and Sanjana. It's now time for the question and answer session. To ask a question, please type it in where it says type your questions here and then press submit. So our first question today asks, can you explain more about the surfacome library you used for the functional profiling work? Is it CRISPR or RNAi, and does it have barcodes too? Also, what genes are you targeting? Is it just surface proteins?
Yes, thank you for the question. So the first library that we applied was an RNAi-based library using barcodes. Essentially what we did was we built a custom library targeting specifically surface proteins that either contain transmembrane domains, GPI anchors, or have evidence of mislocalization in cancers. And this was essentially prioritized using 3 different data sets. First, the SILAC data set, which is at the protein level, seeing essentially or identifying surface proteins dependent on mutant RAS expression. We also apply data sets looking at differential expression with matched normal tumors, and then refining that with microdissected tumor data sets to make sure that we weren't picking up stromal hits. And we also looked at copy number amplification trends as well. So this is a multifactorial data set that built this library.
Thank you very much. Our next question asks, what are the therapeutical consequences of the different subtypes?
Thank you for that question. So while we're not exactly sure of what the therapeutical consequences are at this time, what we can say looking at our single cell feature barcoding data is that the classical and basal groups are responsive to different types of knockout. So to extend off of that, they would be responsive to different kinds of drugs. So usually when tumors are treated, or pancreatic tumors are treated with chemotherapy, there seems to be some sort of a remission, and when they relapse, these tumors relapse as a completely basal phenotype, suggesting that only the classical and perhaps quasi-basal subtype are responsive to chemo. So while we're not exactly, while we can't say right now what the exact targets would be for each of these subtypes, they do definitely respond to different treatments. So there would need to be some sort of either sequential or combinatorial therapeutics to target both subtypes.
Great. Thank you very much for that. A quick reminder to our live audience, you still have time to ask a question. To do so, please type your question in where it says type your questions here, and then press submit. Our next question asks, does driver map use molecular barcodes to protect against bias amplification?
Yes, that would be for the driver map technology. We actually looked at that, but we don't include them in the driver map approach, and there's a couple reasons. The one thing is it does complicate the analysis a lot, and it doesn't necessarily seem to provide as much protection against bias as you might think theoretically. But also, if you think about qRT-PCR, really there aren't usually the barcodes included there in the qRT-PCR, and it's really more important than to maintain sort of a reasonable cycle number. And the other approach is it would require then with the barcode deeper sequencing, and one of the things that we wanted to do was have an approach that you could look at a large number of samples with, without going so deep in terms of the sequencing. So we basically chose to keep this system more simple and straightforward than overly complicating it.
Okay, great. Thank you very much. Next question. What is the relevance of using pancreatic cell lines versus PDX model for CRISPR screening, or in vitro versus in vivo screening, preferably in the orthotopic model? Who would like to take that, please?
I can take that. Comparing cell lines, classical cell lines, to cell lines derived from our PDX models, we find that we do see an increase in the amount of intra-tumoral heterogeneity within these models, which was originally identified in Andrea Viale's group, which was at the end of the Selecta presentation. We identified subclones that were actually responding with opposite phenotypes. So on one hand, these PDX models have more intratumoral heterogeneity, which lets us sort of explore that component. And on the other, these PDX lines are also derived from the same cohort that we built the network with. So the idea was we wanted to sort of bridge that gap. And even though it's still a secondary transplant orthotopically, when we do these in vivo screens, and we've shown through competition assays, biophysical competition assays, that we can identify specific in vivo hits that you would never see in vitro. But what really the importance there is that comparing in vivo and in vitro isn't about identifying hits that only score in vitro or only in vivo. If we're using essentiality as a way to sort of additional way to characterize our models, what's more important is the rank or spectrum of these dependencies in defining these models. Because when you go in vitro versus in vivo, you may have a few similar hits here and there, but the rank is going to be dramatically different, and that's going to have an effect on how we interpret the data as well.
Thank you very much for clarifying that. Very helpful. A quick reminder to our live audience, you still have time to ask a question. To do so, please type it in where it says type your questions here, and then press submit. Our next question then, is the quasi-basal tumor characterization a tumor that is in transition from classical to basal-like subtypes, or is it distinct from either type?
So I can answer that. So it's a little bit of both. So what our hypothesis is that the quasi-basal state is a transitionary state where a tumor is kind of losing its dependence on a lot of the classical markers and gaining dependence on basal markers. But we also hypothesized that the quasi-basal state is a plastic state, and it's the sort of the most comfortable space for a tumor to be, where it can take advantage of both spectrums. So while it is a transitionary state, we do think that it is a distinct and stable state that isn't necessarily going to transition all the way to basal unless there's a clear selection pressure to do so.
Okay, thank you very much. Sanjana, a reminder to our live audience, you still have time to ask a question. To do so, please type it in where it says type your questions here and then press submit. Our next question. How did you do the expression profiling of the 50-some PDX models? Is it standard RNA-seq?
So yes, it is standard RNA-seq. So how we did the profiling for these models is we got the patient sample. So basically the patient tumors were dissociated, grown in a mouse, and then we did RNA sequencing of the entire tumor chunk, and then did aligned it with both the mouse and human genome, and subtracted any reads that came from the mouse genome to remove any mouse infiltration and also stromal infiltration.
Great, thank you so much. And that is where we are going to finish for today. I'd like to thank Sanjana Srinivasan, Johnathon Rose, and Dr. Paul Deal once again for their presentation and for answering your questions. I would also like to thank the webcast sponsor, Selecta, and of course you, the audience, for taking the time to be with us today. Remember, you can watch this webcast again at any time on demand at nature.com/webcasts. Thanks for watching, and I hope you'll join us again soon.